Hello and welcome, fellow biochemists, to our BIS103 Lecture 11. Today we'll talk about the electron transport chain, or the ETC. So at long last, it took us 10 lectures, but today we will actually start making ATP. So all our hard work will start to pay off. To start off with, just to highlight, I've broken up this lecture again in 2 videos that will focus on slightly different aspects, so watch out for that. So let's start with our ETC. For nomenclature purposes, to begin with, there are different names again for this pathway that you may find in different textbooks. For the most part, you will see it as the electron transport chain. Other names are also the respiratory chain. We'll see where this name comes about, and also called oxidative phosphorylation. And we'll learn in the next lecture, in lecture 12, a bit more what this has to do with phosphorylation. But you can also imagine it, right? We're talking about ATP, so it makes sense to think about phosphorylation. So for this video, our major learning goals are 2. The first one is that we want to be able to explain the composition of the complexes and the enzymes that are part of the ETC and how these complexes are important for the transfer of electrons and protons. And in addition, we want to be able to describe the different carrier molecules that we're using to run our ATC. To begin with, let's sort of bring everyone onto the same point. Before we want to start the ETC, we have to remind ourselves and look back at all the reduced electron carriers that we have generated so far in BIS103. So these are our NADH and our FADH2 molecules that we have generated through a number of our oxidation reduction reactions in various pathways, and they now want to use essentially as our currency to make ATP. So if you think back about some of our major pathways, right? We had talked about our glycolytic pathway breaking down glucose as well as other carbohydrates, as well as our polysaccharides down to pyruvate. And then in presence of oxygen, the PDH can convert pyruvate into acetyl-CoA. Acetyl-CoA will then go through the TCA cycle. We also had already talked about, right, our fatty acid beta oxidation that allows us to break down long-chain fatty acid into acetyl-CoA. And in a few lectures' time, we'll also see that also amino acid breakdown actually releases acetyl-CoA. So in all these reactions, right, what we have done is we have broken down these more complex metabolites. And part of these reactions, the importance was to generate these reduced electron carriers. And so the major part that we discussed was all the NADH and the FADH2 that we're generating in the decarboxylation of acetyl-CoA to generate NADH and FADH2 in the TCA cycle. And that now we want to use for the ETC. But we have to keep in mind, right, these electron carriers also came from a number of other pathways of beta oxidation, right? If you're looking back to that lecture, we also there had generated NADH and FADH2. Also those go into the ETC. Our PDH reaction releases NADH in the oxidation of pyruvate to acetyl-CoA. Our reaction 6 in glycolysis, right, the reaction from glyceraldehyde 3-phosphate to 1,3-bis-PGA is releasing NADH. And we will also see that also again, amino acid breakdown is releasing NADH. So we're now sort of pooling all our money from all these pathways together, and we want to use all the electrons and the energy that is trapped in these electrons and use them for the ETC to ultimately make ATP. So the point is, right, we are driving ATP production from ADP. This is our substrate here with all these electron carriers. And please keep in mind, this occurs only in the presence of oxygen. So let's start with a bit of an overview. What actually is the ETC? The ETC actually is a series of electron transfers. Electron transfers, right? If you think about biochemistry, these are oxidation reduction reactions. So we're actually not learning too much more here in terms of biochemical reactions. Mostly we are dealing with the oxidation reduction reactions that we have already seen all quarter. Much like those pathways, those oxidation reduction reactions are catalyzed by enzyme complexes. They're a bit different here. They're actually made up of proteins and these electron carriers. There are four major complexes that we need to look at for the ETC. Our complex one here is the NADH dehydrogenase. The name already tells us it is catalyzing an oxidation reduction reaction. The second one is complex 2 here, the succinate dehydrogenase. This one should ring a bell. That's actually an enzyme of the TCA cycle. So this enzyme is doing double duty. It's functioning in the TCA cycle and as complex 2 in the ETC. Then our complex 3 here. Now we're going into enzyme complexes we haven't really heard about before. We're going into our cytochromes. So the cytochrome BC1 complex is called for short complex 3. And then the last one here, the complex 4, is the cytochrome oxidase. These are our four major players that are partaking here in the ETC. There are two more that we need to pay attention to. There are actually two more dehydrogenases, the acyl-CoA dehydrogenase and the glycerol-3-phosphate dehydrogenase. They also are feeding in reduced electron carriers in addition to these four complexes. We'll see how that works too. And then ultimately, the last complex in the ETC actually is our complex 4 here. What complex 4 does, it actually uses oxygen as the last terminal electron acceptor. And what happens here, upon this reaction, we are generating water, H2O. This is what we call respiration. This is why you have to breathe, why I have to breathe, is to run our ETC, because we need that oxygen that we're breathing in as a terminal electron acceptor. Without oxygen, we cannot do the ETC. Another important aspect to think about when we want to talk about the ETC is that these complexes are, for the largest part, membrane bound. And they're bound at the inner mitochondrial membrane when we talk about eukaryotes, and they're bound to the plasma membrane when we talk about prokaryotes. The examples I'll be using today will focus on the eukaryotes. The biochemical principles are essentially the same, but we want to focus here on the mitochondrial membrane for the purpose of this class. These enzymes being in the membrane, being bound there, is really important because the purpose really is to use the electron transfer in the ETC and couple it to the transfer of protons across this membrane and across a gradient of protons. So this is hard work to get something across a membrane against a gradient. So we're using the energy in the transfer of these electrons to do this. And we see how this works. And ultimately, doing this, pushing these protons across the membrane, this will create what we call a proton motive force, or PMF. We'll see a little bit later how this works. But this proton motive force, actually the release of this energy, this is what enables our biochemical work. And here for our ATP production, this biochemical work is the ATP synthase that actually forms ATP from ADP. So now just to illustrate this concept in an image, right here you have your membrane looking at the eukaryotes here. So this here mitochondrial inner membrane right here. On the inside, this is now the inside here, the mitochondrial matrix. And on the outside here, we have the intermembrane space, right? So this is the space between the inner membrane and the outer membrane of the double membrane of the mitochondria. This is where we are. And so your complexes now are either integrally embedded into this membrane, or at least they are bound to it. And so they're nice and tight sitting here in this membrane so they can facilitate these electron transfer reactions. And so just to continue orienting ourselves right now, we're bringing in our reduced electron carriers here, our NADH and our FADH2. We're bringing them in and basically handing over the electrons to these different complexes and have them run through a series of oxidation reduction reactions here through these complexes. Right, so NADH gives up its electron, it's giving it over. Same happens here, and then the electrons continue to flow through these complexes, ultimately ending up at oxygen. We're generating water. And so at the same time, as we are moving these electrons along, we are pushing protons across this membrane into the inner membrane space. And the backflow of these protons through the ATP synthase here that I highlight just here, that actually is what allows us and gives us the energy to make ATP. It's a really cool system. It's biochemically absolutely intriguing, right? All right, so this is what we want to achieve, making ATP, using the energy from transferring electrons across these complexes, while at the same time pushing protons across the inner membrane. So if we want to do this, right, it seems worthwhile that we actually have a look at our electron carriers that we can use and that partake in our ETC. Right? There are a number of those, and we need to be familiar with those if we want to understand how this works. Two you're already familiar with, right? Our NADH, we talked about it a lot. What I want you to remember for the ETC here is whether or not they're soluble or enzyme bound and how many electrons they can carry. Right? So for NADH, this is our major one that we will be discussing here. It's an obligatory 2-electron carrier. It always has to carry 2 electrons, and it is soluble. Its body here, FADH2, is different, right? It also can carry 2 electrons, but it can do so in parts. So it can be 1 plus 1. So it can be a 1 or a 2-electron carrier, and it is enzyme bound. It does not actually exist in a soluble form. It's always bound to the enzyme. So those two we already have covered sufficiently in the previous lectures, but there are a few more that we want to look at. The first one up here is our ubiquinone. There are several abbreviations that you might find in the literature. The most common one here is CoQ or CoQH2 in its reduced form, CoQ in its oxidized form. You also find it as UQ or UB. For the purpose of our course, I will abbreviate it as CoQ. Up here, you see its structure. You do not have to memorize it by any means. But what you see is, right, is a very long hydrocarbon tail here. This is very hydrophobic, and it allows it to very easily interact with membranes. So it's important for its function, the ETC. The hydrophobicity allows it to move within membranes. And then the business end is right here. And I've just sort of zoomed into the business end down here. So here's the oxidized form, which is a quinone. And you can see right here, the oxidized form are these 2 carbonyl groups that are bound in the ring. Now these can be reduced to hydroxy groups right here. This is a reduced form now, a quinol. And so much like FADH2, this can transfer 2 electrons, but it can again do so 1 at a time. And that's really important. We'll see this later for its function in the ETC, that it has this versatility of being able to carry either 1 electron at a time or also 2 directly. Okay. An additional importance for CoQ is that it also carries protons, and so it actually contributes to the coupling of the electron transfer with the movement of protons across the membrane. And maybe its most important role as an electron carrier is that because of this hydrophobic tail, it is very mobile and it's protein free, so it's very small. And because of this, it can move quite easily in the membrane, and it can actually move between all the other complexes. And we'll see why this is really important. And so let's have a look at this, right? So I'm trying to reorient ourselves now back into this entire ETC series of complexes, and your CoQ is sitting right here. So it's sitting sort of between our complexes 1, 2, and 3 right here. Okay. And now because it's such a small hydrophobic molecule, it can move around here in this membrane and can actually be in contact with each of these different complexes. And so what that allows us to do is that we are branching our electron transfer chain. Okay. So it's not just a series of events from complex 1, 2, 3 to 4. Actually, CoQ branches the ETC by being able to interact either with complex 1 or with complex 2 here. Okay. So this is very important if you think about a fail-safe mechanism of bringing in electron carriers in the reduced form into the ETC. In the next video, when we talk about some of the approaches that we can use to study the ETC, we'll go into a bit more detail of why this is so critical. So in a way, you can sort of see CoQ as a pizza delivery guy, right? It's sort of going always back to the pizza store, is grabbing more pizza, and can move them around between these complexes here and deliver electrons, in this case, between the complexes. So just as a little sort of memory help, you can think of it as a pizza delivery guy for our ETC. Right, the next carrier, we're not quite done yet. The next carrier we actually see are iron sulfur groups, or also called non-heme irons, but mostly you can remember them as iron sulfur groups. There are many of these, and I sort of have some schemes right down here. The key part is this iron atom in the middle. Okay? And so this iron atom in the protein, they're always protein bound electron carriers. The part of the cysteines here, that these sulfhydryl groups of the cysteine amino acids are coordinating the iron atoms and keep them in place. The difference here is simply the number of irons you have in these complexes. Their function, however, regardless of what the structure really looks like within the protein, is that they are a 1 electron carrier and a 1 electron carrier only. So they will always only carry 1 electron, and they are protein bound, much like we had seen for FADH2. Just to sort of give you a sense of perspective what this looks like, here are two iron sulfur clusters, here an iron 2 sulfur 2 right here that is sitting bound in this small protein here. So they're complexed with this protein. Here's a bigger one, an iron 4 sulfur 4 complex sitting right here bound to the protein, just to sort of give you a sense of what it actually looks like in the protein complex. Next one up, very similar concept, cytochromes. And so cytochromes are actually a combination, a complex of a heme and an apocytochromium protein. Okay. So much like our iron sulfur clusters, these hemes are always bound to a protein. What they look like is they are these larger porphyrin rings here that coordinate in the middle again an iron. So the biochemical principle is actually almost exactly the same as with our iron sulfur clusters, only that we're using a different substructure here to coordinate the iron. But the idea is the same. You can have the iron here in its oxidized state, and it can transfer 1 electron, reducing the iron in the middle here. Okay. So much like the iron sulfur clusters, the hemes are obligatory 1 electron carriers. They're very, very abundant. Again, just to give you a sense of perspective here, this is cytochrome C, one of these sort of semi-mobile proteins in the ETC, and you see the heme here coordinated right in the middle of this protein. Cytochromes are extremely widely distributed. They're used for a variety of different purposes, far, far beyond just the ETC. We won't be able to touch on any of these really throughout the quarter, but just keep in mind these cytochromes are really very abundant. Often organisms have hundreds of these proteins. Just sort of one prominent example is actually your hemoglobin, right? So the protein complex that actually allows your red blood cells to transport oxygen. And so hemoglobin is actually a globin protein here with these hemes coordinated within the protein subdomains and carrying this iron atom in the middle. That iron atom is actually what binds, in this case, your oxygen. And because of this, because of the iron, hemes actually are red. And this is what gives your red blood cells the red color. It's actually the heme that does that. All right, last but not least, copper. We can actually also use copper atoms bound to proteins as 1 electron carriers. And again, for sort of just a sense of perspective, these are a bit less sophisticated structurally maybe, but they do the trick. They're not quite as widely distributed and abundant, but essentially you just have a protein here which coordinates these 2 copper atoms here by using different functional groups of a number of different amino acids that keep the copper atoms in place within the protein. So these are your electron carriers. These are the ones that actually are all playing roles in these electron transfer oxidation reduction reactions in your ETC. So now that we understand who's in the game, who are our players, we want to also understand where it is happening. Right? The ETC is located in the mitochondria. We already mentioned it is located at the mitochondrial inner membrane. So right here you have your mitochondrium. You have your outer membrane here, sitting here. That actually is pretty much permeable for all small molecules as well as ions. So that's not really much of an issue for us. Everything can get through there. And then the inner membrane is sitting right here, and you see it has these invaginations. I already mentioned that earlier in our lectures, that basically help to increase the surface area within this mitochondrium here, so that we can pack more of these ETC complexes in one mitochondrium, so we have more capacity to do ATC and to do ATP synthesis. But these complexes now are all embedded in this inner membrane here. So we have right here our carriers and our complexes and our ATP synthase, they're sitting right there. And then in the matrix, so in the soluble fraction that is sitting in between now, of the inside of the inner membrane right here, we have a lot of our other enzymes and pathways that we had talked before. Like we have our PDH complex that's in the matrix, it's soluble. We have our TCA cycle enzymes in the matrix here. We have our beta oxidation enzymes sitting here and our amino acid oxidation enzymes too. So the vast majority of enzymes and pathways that are now producing our reduced electron carriers that we need for the ETC are all located in the matrix here, so have direct access to the ETC at the inner membrane. The one big exception is glycolysis. Glycolysis, right, is in the cytosol. So we have to think about, well, right, in glycolysis, we have our reaction 6 that is generating NADH that can be used for the ETC, so we don't want to lose out on it. So we have to have a way on bringing it in to the mitochondria. Again, right, I said the outer membrane is permeable, so that's not an issue, but the inner membrane is not. So we have to have an active way of bringing in the NADH from, for example, glycolysis, as well as a number of other cytosolic reactions that we didn't touch on throughout this course. So how do we do this? Right? How do we get all of our electron carriers into the matrix? So we have to look at our shuttles. Right? We've touched on shuttles already a couple of times as a means to transfer electrons or cofactors across the membrane without disturbing the individual pools of cofactors, for example, between compartments. And so we can help ourselves with these metabolic shuttles also for this purpose here. And there are two shuttles that are of particular importance for doing this. The first one you already have heard of several times now. That's our malate shuttle, right? We had already encountered this in G&G as well as in fatty acid synthesis, where we wanted to get things from the mitochondria into the cytosol. So we have understood already how the malate shuttle works, but we've used it in the other direction. Today, we actually want to start here, and we're going into the mitochondria, right? So assume you have made NADH in your glycolytic pathway, right? So we can use this reaction, we can convert it over, and we can use malate to bring it over here. Now malate can be oxidized again to OAA, and we're releasing NADH on the other side. Okay. So we now have brought in our NADH from the cytosol into the mitochondria. We have achieved that. We leave it at here for now. There obviously is also a way to bring it back because, right, we now have the carbons of OAA sitting here from the oxidation of malate, from the release of NADH. So we need to get those carbons back into the cytosol. And there are actually a number of different shuttles that we can use, but we'll touch on those in more detail when we start talking about amino acids. But this is our major shuttle that we'll be using to bring in NADH as a product of glycolysis, so we don't lose out on it for making ATP. There's another shuttle. It is not as important, if you will. It is only found in certain tissues and active under certain circumstances, but still worthwhile mentioning as an alternate way on how we can utilize our NADH from glycolysis or other pathways actually. And so what we're doing here is that we're actually using DHAP, right, our glycolytic intermediate. And keep in mind that, for example, fructose catabolism ends up in DHAP. So this is actually a feasible shuttle we could use in this circumstance. And then DHAP we can actually use this, and we can use here this glycerol-3-phosphate dehydrogenase, so another enzyme catalyzing oxidation reduction reactions. What we do first is actually we take our NADH from the cytosol and we oxidize it. And then what we do is actually now we have the electron trapped here in this molecule, glycerol-3-phosphate. And then we actually have an isozyme of this glycerol-3-phosphate dehydrogenase that is sitting at the outside of the inner membrane here, right? So we can get that molecule through the outer membrane that's permeable. We need to get it all the way through. But what we want to do is, right, we want to get the electrons from NADH. That's actually what we're really after. So the dehydrogenase can act again. And now it's doing something different. It will actually take the electrons from this molecule that is currently carrying the electrons from NADH, and it can transfer them over to another cofactor, FAD, generating FADH2. And this actually can directly now interact with our CoQ, right, with our pizza delivery guy for the ETC that can take on the electrons from FADH2, and we have brought them into the ETC. So it's nice little alternate pathway of doing this. It has one drawback, right? It has less ATP yield. If you think about bringing in NADH through the malate shuttle, you bring it in and the equivalent is NADH. So you're not losing out on anything, right? Every NADH is making 2.5 ATP. Here, you're actually interconverting it to FADH2. And because FADH2 is directly giving it to CoQ, it will actually bypass one step of the ETC, which means that it makes less ATP. It makes only 1.5 ATP. So we're losing out on 1 ATP for each time we're actually using this shuttle here over the malate shuttle. All right, so now we have brought in all our reduced electron carriers. We can finally do the ETC, right? We have all our money in the bag and we can get going. So what we want to do now is to run it through our complexes, transfer the electrons to generate this proton motive force, to couple it with the transfer of protons. So let's walk through how this actually works, right? So here are your 4 complexes. Here's number 1 on the left. And then number 2 here. Apologies. Let's bring this back up. Here we go. So we have our complex 1 here, then to the right, complex 2. We're moving on, there's CoQ here. Then we have complex 3 and complex 4. And the black arrows here are indicating how the electrons can flow. And you can see, right, that they're all flowing from complex 1 and 2 to CoQ here. We also see our shuttles, right? The glycerol-3-phosphate shuttle is bringing in electrons to CoQ, and then we have our 2 additional enzymes here. Also the fatty acyl-CoA dehydrogenase I mentioned in the beginning that is bringing in now FADH2 electrons from beta oxidation. And so our electron transfer will go from left to right across these complexes. And so each of these complexes is catalyzing oxidation reactions, and you can see them down here. Right? Our complex 1 is taking NADH, and that's the only thing that it's taking. It's not taking FH2, and it will give them over to CoQ. Oxidize NADH, reduces CoQ, that then can give the electrons over. Complex 2 will take succinate, hand the electrons over to CoQ, will generate fumarate as the oxidation product of succinate, and will reduce CoQ, and so forth. So you can move through all these reactions that I have outlined here to really understand who is giving the electrons to whom. Okay. But in this process now, as we're moving these electrons, we're also pumping protons. And we're doing, though, in complex 1, you can see here again by these arrows, Complex 1 is pumping 4 protons while it is transferring these electrons. Complex 3 also is pumping 4 protons, and complex 4 is pumping 2 protons. And so we'll see why this difference exists, as well as why, for example, complex 2 is not pumping protons when we talk about the energetics of this system. And so here's just, again, a summary of all these complexes. I by no means ask you to really remember all of these details here. What I want you to remember is how these complexes work in conjunction with each other, who is transferring electrons to whom. I just want to highlight here how complex these complexes are. And for example, complex 1 here is comprised of 45 proteins and a whole number of electron carriers here, specifically a whole number of iron sulfur clusters, right? And the same is true for many of these other complexes. So they're incredibly complicated proteins combined with these electron carriers. Now, just to illustrate what this actually looks like in a three-dimensional space, this actually is the three-dimensional structure of complex 1, our NADH dehydrogenase. And so what you see here, right here again, is your inner membrane. Your protein is actually embedded in the membrane, so it's membrane bound. And then up here, we have our NADH coming in, and it is transferring the electrons over. And these are basically jumping now from one electron carrier to the other. There's a number of iron sulfur clusters here that do this. And ultimately, we are reducing ubiquinone, we are reducing our CoQ, transferring the electrons over to that. So in a way, you can imagine it as sort of hopping from stone to stone, right? This guy essentially is your electrons. They're jumping from one electron carrier to the other. So these structures, what they facilitate is that these electron carriers are in close enough proximity to each other for the electrons to jump along all the way to ubiquinone, to CoQ, without having them getting lost within the protein. Here another example now, our complex 2, our succinate dehydrogenase. In this case, right, taking on succinate here, we're oxidizing it to fumarate. The electrons are going to our enzyme-bound FADA, we're generating the reduced form, FADH2, and then again, jumping of electrons through these iron sulfur clusters all the way to CoQ. Just to sort of highlight how this actually looks like within a protein. And then both, right, complex 1 and complex 2, what they're doing is they're shuffling the electrons over to CoQ. And so CoQ now again, right, is this small molecule with this long hydrophobic tail, so it can move around here in the membrane. It can grab electrons from complex 1 here. It can grab electrons from complex 2. And what it does actually now is it will transfer all of the electrons, regardless of from where it got it, to our complex 3 here. So this is sort of central station for the ETC. All the electrons that are coming from the two complexes, as well as from the other dehydrogenases that I had showed you, will be given to complex 3. And then complex 3 will actually transfer them over to cytochrome C right here, one at a time, right? Remember, cytochrome C has a heme as an electron carrier. That's an obligatory one electron carrier. So we do it one at a time, but they will be transferred now from complex 3 via cytochrome C to complex 4. So the two complexes are really integrally bound in the membrane. Cytochrome C is attached to the membrane, but can move fairly freely, not as free as CoQ, but fairly free. And then complex 4 will end up with all the electrons and will eventually give them over to oxygen. Right? We're reducing it to water. We have finalized our transfer of electrons. So this is the end of this video. I wanted to highlight for you how the mechanism of electron transfer works. What we will actually now look at in the next video is we'll look at how the energetics work, how we're actually generating energy as part of this transfer of electrons to bring about the transfer of protons, as well as to bring about eventually the ATP synthesis. And we'll also look at some experimental approaches that people have used and they're still being used to study and to understand how the ETC actually works.